Good afternoon. Welcome to our weekly Sano seminar. Those of you who are surprised that Marian is not present, he is unfortunately not available today, and I will try to make a replacement. My name is Tomek Gubała. I'm one of the co-founder of Sano, the International Institute for Computational Science, Computational Medicine in particular here in Krakow. And today we are happy to have with us Dominika Ciupek, who has agreed to present a very interesting topic for our today's seminar. But before I let Dominika start, we also have with us Professor Tomasz Pięciak from the University of Valladolid, who is Dominika's supervisor and who would like to introduce Dominika's track record, scientific track record. So Tomasz, please switch on and tell us something.
Could you hear me correctly?
Yes, very well.
Good afternoon to everyone. So welcome to today's session. Today's seminar is about deep learning, about the fascinating topic that is about how to join the data from heterogeneous sources. I mean, typically in deep learning, you use some data from some kind of sources for training, for evaluation. However, recently it has been observed a trend to merge the data from different sources, not only sources, but also different modalities, different kind of modality setup. So we can maybe we can have one modality like MRI or CT, but in different source that might be different scanners, different acquisition protocols, different protocols to process the data. And a recent topic that arises in deep learning and machine intelligence fields is how to join this kind of data to build a comprehensive model to be able to grasp all the features from all the sites into one model, one single model, and how to do it correctly, how to do it properly. So the talk will be given by Dominika Ciupek, a brilliant PhD student at Sano currently. So I think, Dominika, the floor is yours.
So thank you very much. Today I will discuss my research related to my doctoral thesis on the application of federated learning to medical data at large scale. My supervisors are Dr. Tomasz Pięciak from the University of Valladolid and also Dr. Maciej Malawski, a professor at LGH University of Krakow, who is also the director at Sano and the leader of our extreme scale data and computing team. I am pursuing my PhD as an external student, so we have not yet chosen the university where I will defend my thesis. The potential universities are Warsaw University of Technology and the LGH University of Krakow, and maybe also the University of Valladolid, but we will see. And due to the external mode of my PhD, the specific discipline for my PhD is not yet determined, but it will likely be either biomedical engineering or computer science. Right now, I am focusing on three main research problems. The first two are related to federated learning, and the third one to standard deep learning algorithms. The first problem is about the application of federated learning to translate magnetic resonance imaging data with a specific focus on brain tumors. The second one involves the utilization of federated learning to estimate brain microstructural parameters based on the diffusion MRI data. And lastly, the third problem is centered on analyzing the extent of generalization of deep neural networks for estimating brain microstructural parameters based on the MRI data across different groups. And these groups may have varying acquisition parameters and the presence of different diseases such as ADHD or bipolar disorder. And first, let's explain what the federated learning is. Federated learning is a decentralized approach to training machine learning models without sharing the data. Modern machine learning models require large and diverse data sets for training in order to achieve high accuracy. However, creating such data sets, especially with medical data, is a highly challenging process due to two main factors. Medical data is highly sensitive and must be protected, and it's also very doubtful that such a large and diverse data sets would be available for free. In contrast to traditional machine learning methods that collect the data from different institutions on a single server, federated learning transfers only the parameters of locally trained models to generate the global model through aggregation. When implementing the federated learning architecture, there are two main strategies to consider: centralized and fully decentralized. The centralized federated learning requires a central server. It generates the global model based on the local model's parameters and also maintain the communication between the central server and each client. The most popular decentralized approach is the peer-to-peer strategy, and it eliminates the need of a central server and also treats all the participants equally with equal privileges. In this strategy, each client exchanges its own parameters of its local models to generate the global model and also perform its own aggregation. Some basic aggregation algorithms include federated average, federated median, and federated Adam. The federated average is the first aggregation method proposed, and currently it is the most popular one. It involves calculating a weighting average of the local model's parameters to generate the global model, and the weighting factor is calculated based on the number of examples at each client. The federated median generates the global model as the median of local model's parameters. And federated Adam is an adaptive optimization algorithm that improves the convergence of federated learning. Although federated learning is a very promising approach, it still faces several challenges. The first challenge is about the model's performance. In the federated learning architecture, we can often see the decline in the global model's accuracy just after the aggregation. And additionally, the global model can perform well for only some of clients and poorly for others. And this imbalance occurs because a neural network tends to favor one client over the other, especially if that client has much more data than other clients. Another problem is data heterogeneity, and it comes from variations in the data acquisition protocols and also in the absence of standardized data processing techniques. This diverse nature of data can result in issues with the performance of global model due to its inability to convergence. Another problem is the potential presence of malicious clients in a federated learning architecture, and this kind of clients can intentionally or unintentionally, for example, due to some errors in the data, send manipulated models parameters for aggregation and therefore can disrupt the proper functioning of the global model. Another problem is security. Although federated learning provides some security by minimizing the need to exchange the data because data remains on the local servers of each client, with numerous clients in the federated learning system that are unknown, the risks of data leakage through reverse engineering is much higher than in the case of standard deep learning. And the last issue is label deficiency. Federated learning models, just like deep learning models, also require large and labeled data sets for training. Unfortunately, in reality, only some of the institutions have fully labeled data, when most of them have fully unlabeled data or only partially labeled data. And additionally, in federated learning environment, different clients may have data labeled to different degrees. So, for example, some of the clients can have fully labeled data, some fully unlabeled data, and others data labeled to various degrees. And now I will present a brief overview of modern methods that enable us to partially bypass the problems that I mentioned earlier. And all of these methods are described in more detail in my review article that will be published on Archive or bioRxiv this summer. And one way to improve the model's overall performance and to avoid the decrease in the accuracy of global model just after the aggregation is to dynamically assign a weighting factor to each client. And this weighting factor can be calculated based on different metrics, for example, on the loss function value or on the accuracy. And an example is federated cost-weighted averaging, Fed cost W average. And here the weighting factor is calculated not only based on the amount of data at each client, but also on the change in the loss function between two consecutive rounds. One technique for dealing with data heterogeneity is split learning. And this method involves dividing the neural network into two parts, global and local, and only the global parameters are sent to the central server for aggregation. However, this approach results in the absence of the global model because as the results of training, each client obtains its own personalized model. Example of split learning is specificity preserving federated learning algorithm for magnetic resonance image, FedMRI, and it is based on dividing the unit model into two parts, a globally shared encoder to obtain a generalized representation at the global level, and only the encoders are sent to the central server for aggregation. And we also have a client-specific decoder to preserve the domain-specific properties of each client. And also, in addition to a standard loss function, FedMRI employs also a specialized weighted contrastive regularization loss to provide the model with the global recognition ability. The method to address the presence of malicious clients in federated learning system is to just filter out parameters sent by those clients during the aggregation. And an example is the distance-based outlier suppression. And this technique measures the distance between parameters sent by different clients. So it calculates both Euclidean and cosine distances, and those distances are used to calculate the normalized weighting factors that are used then for the aggregation. So basically, the malicious clients will have a much, much lower weighting factor than the normal clients. To enhance the data security in federated learning, we can also use the split learning that I discussed briefly earlier. And this method enables different parts of the model to be stored by different participants. An example is a robust split federated learning, which divides the neural network into three parts: head network, tail network, and body network. The head network and tail network are stored by the client servers and the body network by the computation server. And during the aggregation, the parameters of head network and tail network are averaged by the aggregation server and the parameters of body network by the computation server. One approach to address the challenge of limited labeled data is through semi-supervised learning. However, this method can be applied only when central server has access to a fully labeled data, or when only some of the clients in federated learning have fully unlabeled data. And an example is federated alternate training, which alters the training between annotated and unannotated data. And this method uses annotated silos to fine-tune the global model parameters through labeled data. And then this model is used by the unsupervised silos as the target model to generate pseudo-labels for self-supervised learning. So basically, the model aggregation is done alternately. For a few rounds, the parameters of annotated silos are aggregated, and then for another few rounds, the unannotated parameters of unannotated silos are aggregated. On the first project, I collaborated closely with Janek Fischer, and we were focusing on using the federated learning to translate magnetic resonance imaging data. Magnetic resonance imaging is a powerful tool that allows the study of pathological tissue changes and also provides the information about the natural processes of brain maturation and aging. T1 and T2 weighted images are the most common types of MRI, and for the acquisition, we used the spin echo pulse sequence, which consists of 90 degrees pulse, 100 degrees, 180 degrees pulse, and echo. The time between the middle of the first pulse and the peak of the echo is called the echo time, and the time between two consecutive pulses is called the repetition time. And on spin echo imaging, the echo and repetition times are used to control the image contrast and also the weighting of the magnetic resonance imaging. And the T1 weighted images are favorable for observing structures of the brain, such as white matter or gray matter. And T2 weighted images are often used for locating the tumors. Magnetic resonance imaging research can be time-consuming and also expensive due to the long acquisition time. And to address this issue, researchers have started utilizing the deep neural methods to reduce the scan time. And one such technique is data translation, also called the data synthesis. And it uses an image-to-image neural networks, for example, to generate the T2 weighted images based on the T1 weighted images, and also can provide significant clinical value. And for example, if an image is corrupted due to the motion, it can be replaced by synthesized image. And also claustrophobic patients who had to end their scan prematurely can avoid rescans by just generating the missing modalities. Integrating data from multiple institutions with a varying acquisition parameters is essential to use the deep learning algorithms to translate magnetic resonance image. However, this process raises concerns about maintaining the privacy of sensitive medical data and also the limited ability of neural network to generalize. And federated learning is a very promising approach that can address both of these issues. In our research, we used 4 MRI datasets that contain T1 weighted images and T2 weighted images. And in cases where the data was not pre-processed, we perform scar stripping and also the registration of T2 weighted images to the T1 weighted images spaces. And as you can see in the table, different MRI datasets have a different acquisition parameters, and also consists of data of healthy subjects, HCP-MGH and HCP-Wumin dataset, subjects at various stages of cognitive decline, as is free dataset, and also subjects with gliomas, so Brad's dataset. And therefore, the data was heterogeneous. And to even further increase the data heterogeneity, we select a different number of subjects from each dataset for further analysis. And this varied from 26 to 125. First, we visually evaluated the results of translation T2 weighted images and compared them with the original data. Our research demonstrates that generating synthetic images is challenging, particularly when we are dealing with some pathological changes in the brain, such as gliomas. And while individual aggregation algorithms perform well in the case of healthy subjects, in the case of brain tumors, especially LGG, the area of change is significantly blurred. And this experiment highlights the limitation of federated learning AI algorithms and also proves that among the tested algorithms, the FedMRI produces the best results. In our research, we also compared various federated learning methods with models trained on the single data set. And we used relative error value as the metric. In the case of deep learning, the model's performance quality depends heavily on the dataset on which the model was trained. And the best results are obtained by the model trained on the HCP VUMIN dataset, and the worst by the model trained on the HCP MGH dataset. And also here we can see that most models struggle with properly translating the T2 images with the HCP VUMIN dataset. And our analysis revealed that among the tested federated learning algorithms, the FedMRI produces the best results. And additionally, in the case of Brad's HGG dataset, it even produces better results than in the case of the model strictly trained on this dataset. These findings highlight the importance of federated learning, and the most basic federated learning algorithm, so federated average, achieved comparable results to a standard deep learning algorithms, and by using a slightly more complex method and dedicated to MRI data, we can achieve much better results. And we also check the relative error values when translating the T1 weighted images. And the overall relative error is lower than in the case of the previous analysis, but it is still evident that the training dataset greatly influences the deep learning models. And here, even the simplest federated learning algorithm outperforms the standard deep learning methods. In my second project, I am concentrating on using federated learning to estimate brain microstructural parameters based on the DMRI data. So first, let's talk a little bit about diffusion magnetic resonance imaging. The diffusion-weighted sequences, which are based on the post-gradient spin echo technique, are used to acquire the diffusion magnetic resonance signal. And this technique works on a principle similar to the standard spin echo technique that I present earlier. So here we also use the 90 degrees pulse and 180 degrees pulse. And in this technique, additionally, two symmetric strong diffusion sensitizing gradients are applied on both sides of 180 degrees pulse. And if the water molecules are stationary, the diffusion gradients don't affect the acquired signal value. But when molecules are diffunding, we can observe the loss in the signal value. The B value is a factor that reflects the strength and timing of diffusion gradients. So the higher the B value, the stronger the diffusion effects. And in order to generate a field dataset, first the acquisition of B0 baseline images is performed. And for this, we are using the post-gradient spin echo technique, but with the diffusion gradients turned off. And next, the same technique is used, but already with the diffusion gradients turned on. And the value of the signal after applying the diffusion gradients can be approximated by this relation. Diffusion magnetic resonance imaging is a powerful tool for analyzing the microstructure of the brain. However, it is only an indirect method. So getting specific information about the tissue requires using the proper modeling to interpret the diffusion signal. And we can use signal representations of or multi-compartment biophysical models. The signal representations describe the diffusion signal in a voxel without making any assumptions about the tissue. So they can be used for any type of tissue, healthy or pathological, but the estimated parameters are not specific. One of the most widely used signal representation is the diffusion tensor imaging. The diffusion tensor describes the diffusion of water molecules using the Gaussian distribution model, and it is represented as a symmetric positive definite matrix, which has 3 mutually perpendicular eigenvectors and 3 positive eigenvalues. The principal eigenvector determines the main direction of diffusion, so the direction in which the diffusion is the fastest. The eigenvalues determine the diffusion coefficients in each of the three principal directions. And schematically, we can present the diffusion tensor as an ellipsoid. And the major axis of this ellipsoid is parallel to the principal eigenvector, and other axes represent the other eigenvectors. And the length of this axis is proportional to eigenvalues. Now, the multi-compartment biophysical models are increasingly used, and they adopt a schematic tissue geometry, making it possible to estimate relevant microstructural parameters directly. And these models assume that the total diffusion-weighted magnetic resonance imaging signal comes from 2 or 3 non-interchangeable compartments, and each of these compartments is weighted by its relative volume fraction. The intracellular space is most often modeled using the axially symmetrical and constrained geometric models, while the extracellular space is typically modeled as an anisotropic Gaussian diffusion, also called the Zeppelin. The most popular biophysical model is the NODI model, and NODI adopts a tissue model that distinguishes 3 types of microstructural environment: non-tissue compartment, also called isotropic compartment, intraneurite or intracellular compartment, and extraneurite or extracellular compartment. The non-tissue compartment represents the space occupied by the cerebrospinal fluid, and therefore is modeled using isotropic Gaussian diffusion, so as a sphere. The intracellular compartment refers to the space bounded by the neurite membranes and is modeled as a set of sticks. And the orientation distribution of sticks can vary from highly parallel to highly dispersed. And finally, our last compartment, the extraneurite compartment, refers to the space around the neurites, and this space is occupied by various types of glial cells, and in the gray matter also by the body of neurons. And it is modeled as an anisotropic Gaussian diffusion, so as a Zeppelin. The microstructural parameters obtained after modeling the diffusion signal include, for example, fractional anisotropy, generalized fractional anisotropy, mean diffusivity, axial diffusivity, radial diffusivity, and also return to axis probability. The fractional anisotropy measures the anisotropic diffusion fraction, so the difference between the diffusion tensor ellipsoid and an ideal sphere. The generalized fractional anisotropy is a very similar measure, but to calculate this, we are using the diffusion coefficients from a whole set of discrete directions. And when we are calculating the fractional anisotropy, we are using only the diffusion coefficients corresponding to eigenvectors of a diffusion tensor. The mean diffusivity measures the average molecular motion regardless the tissue directionality. Axial diffusivity is a parallel diffusivity measure, radial diffusivity, the perpendicular diffusivity measure. And lastly, the return to axis probability reflects the presence of restrictive barriers in the axial orientation. Estimating microstructural parameters for more advanced models can be time-consuming, and also some of the models require very specific acquisition protocols, and therefore they are useless for clinical data. And this is why using deep learning is desirable. However, there are still concerns about maintaining the privacy of medical data when creating this large data set, and also about the limited ability of neural networks to generalize, so the same concerns as in the case of data translation. And in our analysis, we used 4 different MRI datasets that varied in the term of the scanner used and also acquisition protocols. And although here we have a different number of subjects for each dataset, the number of slices, so the number of true data, was the same for each dataset for each client. And already in the case of visual evaluation of the results, we can observe the similar trends that in the case of data translation. And in the case of deep learning, the model's performance heavily relies on the training dataset. And the worst level of generalization has a neural network trained on the ZJU dataset. And that's because the ZJU dataset is a very specific one, because it contains only data of 3 patients, and each patient was scanned 10 times. And here, the standard deep learning algorithms outperform the basic federated learning methods, such as federated average, federated median, and even federated cost W average. And among tested algorithms, only the FedMRI yields better results. And the quantitative analysis supports the results of the qualitative analysis. The relative error value for fractional anisotropy is similar for basic federated learning techniques and also deep learning. And here the FedMRI produces a slightly better result, and they are not as significant as in the case of data translation. And we also investigate the relative error values for mean diffusivity. And here, none of the federated learning algorithms outperform standard deep learning. And still, FedMRI produces the best results, but the relative error value is higher than in the case of the model trained on the CAMCAN data and Magdeburg data. And the following research was presented during the Neuronus 2024 Neuroscience Forum in the form of a poster. And it attracted significant interest, especially from biologists and chemists that were not familiar with microstructural parameters. And they were very curious how to calculate them and express the willingness to incorporate them in their own research. And finally, let's talk about articles that we have already published or plan to publish. We don't have any articles on the deep learning and federated learning published yet, but now we are finalizing a review paper on federated learning techniques for medical imaging. And we expect to publish this paper on arXiv or bioRxiv around the holidays. Additionally, we are working on a research article about using the federated learning to translate magnetic resonance images. And we also plan to complete this article by the end of this summer. And in the near future, we plan to start working on an article discussing the generalization level of deep learning models across different datasets with varying acquisition protocols and datasets of both healthy and sick individuals. And if all goes as planned, we expect this article to be ready by the end of this year. Furthermore, we intend to present the results of our research on using federated learning to estimate brain microstructural parameters, but this article is more in the future. And we currently have 2 published articles on different topics. The first one is the summary of diffusion simulated connectivity Disco challenge, in which we achieved 4th place in 2022. And the aim of this challenge was to predict the connectivity of numerical phantoms. The second one is the popular science paper that is already published in the recent issue of the Wszechświat magazine. And it provides a brief overview of techniques used to study age-related changes in the brain, with the main focus on diffusion tractography. And we also have 2 articles under review. The first one summarizes the QuantCon challenge, so the quantitative connectivity through harmonized preprocessing of diffusion MRI challenge. And in this challenge, we also took 4th place, but in 2023. And the goal of this challenge was to harmonize the diffusion magnetic resonance imaging data. The second article I write with the collaboration of Kasia and Krysia from Sano, and we explore the preferences of polls regarding the use of artificial intelligence in the healthcare. And that's all for today. Thank you very much for your attention.
Thank you, Dominika. I believe that we now have time for questions from the audience. Mateusz, can you check if we have some? Oh, I already see our guests raising their hands. Can you, Mateusz, let them speak?
Okay, I don't know who is first on top. So, can you hear me?
Oh,
Yes,
we
we can hear you.
can hear you, but very poorly. So I don't know.
Very poorly. Okay.
Oh, now it's better. I don't know what's going on.
Okay, so I'll try to speak as clear as possible. So with the respect to the different federated learning approaches, do you plan to build your own? And the question would be, what is for you, let's say, an acceptable or a good number in terms of, let's say, a regression coefficient that could be considered safe in terms of an application in a medical environment?
We don't plan now to propose our own method for federated learning. We are planning to incorporate more methods of federated learning into one method. I don't know if you can understand this, but combining few methods into one method, because we do some researches on that and have some results, and the combining of methods can result in a better, for example, relative error values than using a single method. And the question about the values that are safe for using, for example, in real life scenario, is very hard, because we want to have the perfect method. But I think that the method that would be comparable with the best algorithm, that is the best model that is calculated on the global data set, like the data set combined from data set of other institutions, it would be the best for us.
So, but what do the medical people say? What do they claim needs to be the right measure for them?
I don't talk with medical people, so we need to do that before applying any of this method in the real life scenario. But now we sadly not talk with them. And another problem about, for example, calculating the microstructural parameters is that it's not very popular now using with clinical data, because, like I said earlier, calculating these parameters can be time consuming, and also often we need specialized protocols. So now we are also working on making it applicable for this type of data.
Thank you.
Thank you.
Thank you, Olaf. Do we have other people willing to ask Dominika a question? I think we now have Wiesław raising his hand and John. So maybe Wiesław was first. Wiesław.
Yeah, thank you, Tomasz. Yeah, thank you, Monika, for this interesting presentation. Regarding your first application, I don't think so. I would wish any patient who has a brain tumor to have only T1 and T2 weighted scans, because I don't believe they are sufficient
Yeah,
for
you are
any
right.
diagnosis. So why don't you include T1 weighted with contrast, perfusion imaging and spectroscopy imaging? Then your case will be more realistic, and the conclusions which you drawn by comparison different situations might be completely different. What do you think?
That's the great advice, I think. Yeah, you are right. We need to incorporate more modalities into our settings. Now we are using only T1 and T2 weighted images, because most open data sets only has T1 and T2 weighted images. And it's very hard to find publicly available data sets with more modalities. Now we are also planning to add the FLAIR data to our analysis, but maybe we need to add more in the future. Yeah, you are right.
Yeah, so maybe the strategy should be different, because what you have been doing might be of great interest of hospitals, of some clinical societies. I believe if you approach radiological societies, they'll be very happy to use your approach, because it will be very beneficial. So try maybe from Polish Society of Radiology, then European Congress of Radiology, and see what would be the interest.
Yeah, thank you very much for that advice. It's a great one. Yeah, thank you.
Yeah, and regarding the error in your first project, I'm not quite sure, because you are not dealing with tumor type. You are saying you are not doing any segmentation yet.
Yes,
You are
yes.
just having some area affected. What exactly is this error, and why it's so important?
We just want to present some results, like in the values, I would say, not only in the visual way. And we used now we are using the relative value of values, but now we are switching from these values to structural similarity index measure. It was only our preliminary studies. And one more thing, because I don't think I think it's very important that these parameters are calculated only inside the brain, because sometimes in the paper that I write, they calculated these parameters on entire image, and in this image we also have like the background, and the background is nearly often near to zero. So the low error from the background is like lowering our overall error values. But now we are switching from these values to structural similarity index measure.
And also in some project you use term ground truths. How you are getting it? You are producing it, some clinicians from the literature?
Here, the ground truths are the real acquired images, because these data sets have both T1 weighted and T2 weighted images. So these are the real images. And when we are dealing with calculating the microstructural parameters, we are using the parametric method. So we are calculating them from the DTI. I'm using a special Python library to calculate them.
Okay, thank you.
Thank you very much. Thank you very much.
I believe that we now have Jean waiting to ask his question.
Hi, can you hear me?
Yes.
Yeah, great. Thanks for this. Very interesting. I have perhaps as a follow-up comment to what Piotr already said about multimodal. Actually, it's more of a comment or a petition to both you and Tomasz, because very recently I have access, or there's a public data set which contains a large data set of multimodal data in brain tumors. And actually, my next project, or I'm actually starting already, is to use this one for some other purposes. But I do believe that maybe we could chat or organize, because it has, amongst other things, like contrast enhanced, flare, and even diffusion data.
Yeah, that would be great.
Yeah, and of course, because just you mentioned that these microstructural models, they have some problems, of course, and I don't know how they behave even in the presence of a brain tumor, which is like complicates things a lot more. So maybe it's not a bad idea, or it would be worth to exchange some ideas, and I am happy to share this data set if it's of use, of course. Yeah.
Yeah, thank you very much. Yeah, that's a great idea. Yeah, thank you.
Thank you, Jean. Since I don't see more people raising their hand, I have one question to you, Dominika. And excuse me, that question is probably very naive, but so I was thinking about obviously we in one of the main topics of the current debate around the AI in medicine is about overrepresentation of specific groups of people, right? With, for
Yeah.
instance, ethnic or genetic background, that AI models are being trained on, and then these models are not being fully applicable to citizens of other, continents
Yeah.
or countries. But from the technical point of view, and again, excuse me, this is probably a stupid question, but let us imagine that we have a model well trained on, say, NHI open data catalog of MRI images,
Mm-hmm.
and then we have. That's done, and we have another opportunity, for instance, having a hospital from Europe or from Africa joining our network of collaboration, and they also have their own data set,
Mm-hmm.
but they are not willing to donate it to a
Mm-hmm.
central server. From the technical point of view and also performance point of view, is it possible to use already trained model
Mm-hmm.
on our site
Mm-hmm.
in such a federated learning scenario with the other, say, African model being trained in their clinic
Mm-hmm.
and the weights being merged together? Or is it either technically impossible and we have to retrain everything from scratch, or would that simply not make any sense from the performance point of view?
It's not impossible, but I would say that we need more rounds of the training. Not that they can only send us once their parameters of their model and then we are combining it to generate the global model. We must once again set this global model to each of this institution. They must once again train it, and we need to do that for some of the rounds. So this can be a problem.
Are you familiar maybe with techniques in which we would transfer our already trained model to the African site and have that model, say, augmented to generalize also over the African data?
No, sadly, I'm not familiar with such a method.
Okay. Thank you. So
Thank you also.
I believe there are no other people from the audience wanting to ask more questions. So I think that it concludes our meeting today. Once again, thank you, Dominika, for providing us with this interesting presentation of your research. Thank you, Tomasz, for introducing Dominika. And thank you all our guests for attending our Sano seminar today. And I think that's it from me.
And also thank you very much for inviting me here.
You're welcome.